comes the megasporogenesis. Megasporogenesis means formation of megaspore. Formation of megaspore from megaspore mother cell. First of all, what you have to know is what is the other name for megasporangium? Because now this megasporogenesis takes place inside megasporangium. So what is this big word? This big word is actually another name for ovule, which is there inside ovary. Now ovule, since I told you, we have to learn the structure of ovule too. How to draw ovule? How to draw ovule? Draw something like this, like this oval shape. Draw integuments. What is this integuments for? What is the use of integuments?
Protection.
Yes, integuments are there for protection. Now these integuments, from integuments itself I have taken a stalk out. What is this stalk called as?
Funicle.
Yes, this is called as funicle. That's how it is attached to the placenta of ovary. There is a part where no covering of integument is there. What is that area called as?
Micropyle.
Yes, this is called as micropyle. Later, when this ovule will become a seed, this micropyle opening, this area, it allows oxygen and water to enter into and help the seed to germinate. That is later. At present, micropyle will remain like this as an opening where integuments are not covering. Now, there is a tissue here. Like sporogenous tissue is found inside the microsporangium. In the megasporangium, there is a tissue. What is this tissue called as? What is this tissue called as? It is also diploid. It starts with N. What is it?
Nucellus.
Yes, it's called as nucellus. And it will be diploid. Megaspore mother cell will get differentiated from nucellus. That means from the nucellus, one cell will get specialized. That cell is near micropyle. Okay, that cell is near micropyle. And that cell is called as megaspore mother cell. I'm writing in short. It is diploid. This megaspore mother cell will undergo meiosis. When it undergoes meiosis, it will give rise to 4 megaspores. It will give rise to 4 megaspores. Each one will be haploid or diploid?
Haploid.
Yes, very good. Since I told you meiosis, whenever meiosis takes place, they will be haploid. So we can write it as N. But it's not like microspore tetrad. This tetrad, only one will remain, 3 will get degenerated. So in this 4 megaspores are produced. Out of the 4 megaspores, only 1 megaspore will remain functional. And that megaspore is called as functional megaspore. Because of this reason, it is also called as monosporic development. Because there is only one functional megaspore. What will happen to this one functional megaspore? Correct. So that functional megaspore will become the embryo sac. Will become the embryo sac, and here near the, if that embryo sac side is facing micropyle, then you will find the egg apparatus in which the center cell is called egg. Other two cells will be called as, other two parts will be called as synergids. Yes. And I have told you synergids have filiform apparatus also to guide the pollen tube, because future pollen tube will come through this micropyle carrying 2 male gametes. So that should be guided, no? That job is done by filiform apparatus in synergids. At the center, there is a central cell with 2 polar nuclei, but they won't fuse, they are separate. Each cell inside this embryo sac will be haploid only. Now the opposite side of micropyle. What is this opposite pole called as?
Chalazal.
Yes, it is called as chalazal end. That is the opposite side of micropyle. There also the embryo sac will have three cells. Those three cells are called, what is it called as? It faces chalazal end. What is?
And
Yes, great, good. It's called as antipodal cells. So that is the structure of embryo sac. Now when question comes, you have to read whether they're telling you to draw ovule or embryo sac. If they're telling you to draw ovule, draw it, labeling nucellus and other parts. No need to draw this much detail. When they tell you to draw embryo sac, this diagram, what is there here, this much part, this you have to enlarge and draw embryo sac. So that's about megasporogenesis.